As I covered in my previous video on fatty acid metabolism, fatty acid oxidation produces free acetyl-CoA, which may enter the citric acid cycle. In the liver, acetyl-CoA has an alternative fate, which is to undergo conversion to ketone bodies for export to other tissues, such as skeletal muscle and the renal cortex, where they are reconverted to acetyl-CoA and oxidized in the citric acid cycle. First, thiolase condenses two molecules of acetyl-CoA to form acetyl-acetyl-CoA, releasing a free coenzyme A. This step is simply the reverse of the last step of beta oxidation. Next, acetyl-acetyl-CoA condenses with another molecule of acetyl-CoA to form beta-hydroxy-beta-metaglutaryl-CoA, abbreviated as HMG-CoA, which is catalyzed by HMG-CoA synthase, releasing coenzyme A. HMG-CoA is subsequently cleaved by HMG-CoA lyase to form acetyl-CoA and acetyl-acetate. Acetyl-acetate can undergo spontaneous decarboxylation to form acetone. This reaction could also be catalyzed by acetyl-acetate decarboxylase. Acetyl-acetate can also be reduced by beta-hydroxybutyrate dehydrogenase to form beta-hydroxybutyrate, which is coupled to the oxidation of NADH to NAD+. Together, acetyl-acetate, acetone, and beta-hydroxybutyrate are known as the ketone bodies. The rate-limiting step of ketone body biosynthesis is HMG-CoA lyase, because HMG-CoA has an alternative fate, which involves reduction to mevalonate, which is a precursor to cholesterol biosynthesis. Ketone bodies synthesized in the liver are then transported through blood to other tissues, including heart muscle, skeletal muscle, kidney, and brain. The brain prefers glucose as an energy source. However, during starvation, the brain can also adapt to catabolize ketone bodies. During ketone body degradation, beta-hydroxybutyrate is oxidized to acetyl-acetate by the reverse reaction of beta-hydroxybutyrate dehydrogenase. Acetyl-acetate can be directly converted to acetyl-acetyl-CoA by beta-ketoacyl transferase, which transfers coenzyme A from succinyl-CoA to acetyl-acetate, forming succinate and acetyl-acetyl-CoA, which can be reconverted to 2-acetyl-CoA by thiolase. Acetyl-CoA can then enter the citric acid cycle and oxidative phosphorylation to generate energy. Several ketogenic amino acids can also be degraded to ketone bodies. Threonine, isoleucine, and leucine can be degraded to acetyl-CoA, whereas lysine, leucine, phenylalanine, tyrosine, and tryptophan can be degraded as acetyl-acetate. Note that during starvation and untreated diabetes mellitus, glucose is unavailable as a fuel source, leading to an overproduction of ketone bodies. The increased blood levels of acetyl-acetate and beta-hydroxybutyrate lower the blood pH, causing a condition known as acidosis, which can lead to coma and, in some cases, death. Excessive levels of ketone bodies in the blood and urine is known as ketosis. Together, these two conditions are known as ketoacidosis.